So we have here genetic mutations. So let's go ahead and start the slideshow. All right, what are genetic mutations? This may be something you think about when we think of when people say genetic mutations. It's certainly something I think about. Super awesome series of movies, X-Men. However, genetic mutations, and we may talk about them like, oh, I wish my genes can mutate into such and such thing, like I could fly or read people's minds or you know make things super cool or whatever. Well, um, genetic mutations, unfortunately, are usually very bad. So this is not X-Men is obviously not that anyone was thinking it, but it's not a very good portrayal of what genetic mutations are. In reality, genetic mutations are pretty harmful. So they cause diseases and in some cases even death. One very famous um, or popular genetic disorder is Down syndrome. It leads to um, varying varying levels of cognitive development, of impairment of cognitive development. However, this doesn't really affect their, at least I don't think so, the lifespan, but it does affect quality of life, and especially it requires them to have caretakers. Um, then there's also the most lethal, I guess, genetic disorder is called cystic fibrosis, and most of these are this. They're not necessarily cognitive impairments like Down syndrome. Most of them are some type of physical malfunction in the body. Cystic fibrosis deals with the genes, a malfunction in the genes that help regulate the mucus and fluids in your body. And the average age of death for these people is actually 44 years old as of right now. But there's continuous research on improving treatments and genetic engineering for people with cystic fibrosis and other genetic diseases to help improve their quality of life and hopefully eventually cure them from the actual mutation. So genetic mutations are usually harmful, and that's the majority of them right now. However, there's very rare over the course of millions of years, even over the course of millions of years, they're very rare genetic mutations that help improve human life. But that's not. This is usually what we mean by genetic mutations. No researcher or scientist will ever think this. That genetic mutations are beneficial. When you say genetic mutation, their mind will always go to this. This is the most prominent thing right now. But of course, if you're going to talk about millions of years, there may be like every once in a while some rare genetic mutation that nature happens to select for being good at or improving your chances of survival. So that's obviously that, but that's not usually the case. All right, so let's go into types of genetic mutations. What are the actual mutations themselves? Like, what are they, right? What's being mutated? Well, let's go into that right now. It's actually just down to the nucleotide base pairs, or um, longer than the pairs, genes or sections of genes. So, duplication is basically when you have base pairs, like or genes that are duplicated. That's all there is to it. So, if you look at this orange box here to the left and this small duplication example, the DNA, this is double-stranded DNA here. You have four pairs of A's and T, adenines and thymines. But somehow this accidentally got copied one, two, three, four times. Now there are five sections of this A, AT, right? So that can cause some problems in the protein synthesis of this particular gene. Or maybe a whole gene, like here in this second gene duplication example, maybe a whole gene gets copied. So this whole section got copied over here. That could lead to some complications, and or a whole section of so many genes in a chromosome can get copied. So like this one got copied here, so it elongated this chromosome here. So that's duplication. All of these are basically as they sound. Insertion is basically, you have this mutation site here. There's AT on the left, GC on the right of this mutation site, and then an insertion of a new AT gets inserted right there. Or on a macro level, a whole piece of all these genes in a chromosome get inserted into another chromosome, and that can lead to other complications. Inversion is basically just the way it sounds. These genes get detached and then reattached in the inverse manner. And then substitution is when one nucleotide gets substituted with another. So instead of this GC base pair here, it gets substituted with AT. And that might lead to a different amino acid that gets input into the protein down the line, because then it's a different codon here. And different codons lead to different amino acids in the tRNA sequencing. All right, translocation, just like it sounds. It's kind of like insertion, but like more like a switch. The blue got into the orange, and the orange got into the blue. All right, so those are different. Just feel comfortable going through these and just kind of realizing what's happening with the genes or the nucleotides. Very straightforward, nothing complex here. All right, how can we treat some harmful genetic mutations? Well, we can do something called uh, genetic silencing. We can silence genes. So first, before we get into that, let's recall protein synthesis. It involves transcription, which is when an mRNA strand gets developed based off of a particular gene in the DNA. And then that mRNA leaves the nucleus because the DNA cannot leave the nucleus. It's very dangerous for the original copy of DNA to leave the nucleus. Something bad could happen to it, like it could get lost or spliced. And then that mRNA leaves the nucleus and goes to a ribosome that reads this mRNA, and then the translation happens where the ribosome collects a bunch of tRNAs that are reading the codons on the mRNA, and then the tRNAs attach their amino acid that they're carrying around one by one until it creates a big strand of amino acids, and then that once it's finished, the amino acids fold and form a polypeptide, aka protein, this purple blob right here. So genetic mutations can happen during this process. In fact, the mutation for cystic fibrosis is in the gene that helps regulate the protein that regulates the mucus. Sorry, the mutation is in the gene that creates the protein that regulates the mucus. So obviously, that protein will have malfunctions. Now, silencing genes, due to some creative researchers and biologists, they've realized, what if we interrupt this process? That way, the protein never gets created. Like, what if it's a malfunctioning gene that will be not harmful if we just turn it off, per se? And so we can interrupt it in the transcription, and then the protein will never get made, or we can interrupt it in the translation, and the protein will never get made. Well, since it's harder to get to the original DNA, we're going to try and interrupt it in the translation after the mRNA gets created. So how do we do that? Well, there's something called RNA interference. So this is for treatment with people with genetic disorders, and for whom it would not be harmful. It would be good just turning off that particular gene. So we know what the mRNA strand will look like inside the cell. However, and so we create a double-stranded version of it in a lab, okay? And when we create a double-stranded version of it in a lab, the reason we do that is because when we insert it manually into the cell recognizes it, double-stranded RNA, as a virus, a viral RNA, okay? Because that's the way viruses have their DNA. It's just RNA. So there'll be a bunch of things a bunch of proteins inside the cell called RNA-induced silencing complex. They'll come together, they'll break up that double-stranded RNA, and then they'll keep one half of it to start looking for other RNAs that match it. And since we purposely created it to match the mRNA from the DNA that it is going to come out from the DNA, then once the original mRNA that's coming from the DNA comes out, the RNA induced silencing complex will then recognize that original mRNA, and the mRNA will be like, wait, no, guys, I'm the real thing. I'm supposed to create this protein. And then the risk is like, must destroy, right? And so then it just disintegrates this mRNA sequence, which we artificially made to destroy. And so if the mRNA gets disintegrated each time a sequence of it comes out, then the protein will never get made, and it's as if the gene never existed, hence gene silencing. So once again, we create double-stranded version of the mRNA of the complement of the gene we want to silence, and it gets silenced.